Thank you. Thank you for the introduction. Good afternoon, everyone. Thank you for the organizing committee for the invitation to speak today. I want to share with you what we believe is the next breakthrough therapy for CMT1A in the form of Alpha-1 Antitrypsin. So the question is, what is Alpha-1 Antitrypsin? And in short, it's a human protein. It's been discovered in the 1950s as the Alpha-1 fraction of plasma-derived blood samples, and later was assigned the name Alpha-1 Antitrypsin for its main function. And to this day, Alpha-1 Antitrypsin is the name that we use, but in fact, many other positive cellular functions have been attributed to this protein. Immunomodulation, anti-apoptosis, reducing oxidative stress, preventing protein aggregation, and recently anti-inflammation was getting really a lot of attention, but I want to just put the attention in the fact that it's currently being clinically used. So it's an actual approved therapy. So Alpha-1 Antitrypsin inhibits neutrophil elastase, and it is currently prescribed for AAT deficient patients. So these patients, they are unable, due to a genetic mutation, unable to produce enough Alpha-1 Antitrypsin, and this plasma-derived replacement therapy elevates levels of AAT to inhibit neutrophil elastase in the lungs, allowing them to have a normal life. Otherwise, they will be developing a lung disease, which is life-threatening. But I think the key takeaway really from today is that Alpha-1 Antitrypsin has a proven safety profile, has been prescribed for over 35 years, and this is really, I think, the key strength of this therapy. Now, we looked at the component of anti-inflammation, and we did this with a C3 mouse model. Inflammation maybe is looked at as really the cause, not the effect in CMT1A. I think for us as well, it was an attempt to really reduce inflammation or the inflammatory cytokines that are a result of this PMP22 overexpression. And here we took 8 male mice. The C3 mouse model were treated for 2 weeks with clinical grade AAT. And as we expected, this is known that we reduced interleukin-6 levels and TNF-alpha levels. This was not a surprise. What we were happy to learn, that we were actually as well were able to influence the progression of axonal damage as measured by NFL. For us, this was already a positive outcome of the study. What we didn't really expect to see, and this is only after 14 days on treatment, we saw improvement in electrophysiology. So we didn't just stop the progression of the disease, we were actually able to restore the CMAP and NCV levels and increase not only the axon numbers, but also the integrity of the myelin sheath. And this is really the surprise of the study, which we are very excited about. And we wanted to test also, of course, the behavioral tests, so coordination, rotor rod latency, muscle strength, grip strength. So after 14 weeks, both of these parameters were increased significantly, and we wanted to really understand what is the driver behind this link where we reduced inflammation, but at the same time we're able to increase all these parameters. Again, this is not the original design of or the initial aim of the study, but this is surprising and in this significant amount. So what we are focused on now is to really understand the molecular mechanism by which, or the mode of action by which AAT can insert its function in the Schwann cell niche, because really for us the action of AAT is yet unknown exactly, but I just want to share with you a few ideas that we have. And we were lucky enough to collaborate with Michael Serada's lab, where we see that ADAM17, which is a membrane-bound protein, is in the crossroad between inflammation and myelination. So this is, again, preliminary work, but when Michael looked at the rat data that they have for CMT rats, I think for us it was a very encouraging sign, where we see that ADAM17 levels, in fact, stay upregulated. So as these rats develop into adulthood, they have a reduction of ADAM17. In the CMT1A rats, this doesn't go down. So you have really this active ADAM17 doing something. And the question is, what is it? And ADAM17, in fact, is known to regulate myelination. It's known to act through base 1 and neuroregulin 1 type 3. This is, again, also in Michael's work, that this is really in between myelination, and it is also known another name for ADAM17 is, in fact, TACE, which is TNF alpha converting enzyme. So when it is overstimulated, the levels of other cytokines, pro-inflammatory cytokines, are upregulated. So we looked at this RNA sequencing data and thought that this is definitely a direction we want to explore further. So to do that, we worked with plasma-derived AAT, but in order to translate this into a therapeutic approach, we have designed a recombinant version of alpha-1 antitrypsin. And we conjugated 2 AAT arms into IgG Fc domain. This is a common technique to increase half-life in the blood plasma. And this is particularly relevant in a disease like CMT1A, where you might be administering this to a pediatric population. Just to explain that AAT deficient patients have to come in and receive IV every week. That might be very difficult for younger patients, and we believe that by increasing the half-life and also increasing potency, here we take the target of ADAM17, for example, by 18 times, we can really convert this therapeutic strategy from IV to subcutaneous, which allows to open up a therapeutic avenue for this particular design and for AAT in particular for pediatric population. And really, we are just at the beginning of our journey. We came across this discovery, which I'm very, very pleased to share with you today. But I think what we need to do now is really to validate the results that we got, the preliminary results that we've got in the C3 mouse model. So we want to work with, we are working with Michael Sereda to test it in the rat model. We also want to work with Cleopas about testing it in his mouse model. But what is more interesting, and I think this is something that we want to explore more under the Erdera grant framework, is whether this is translatable to other CMT subtypes. And the study in mice and in vivo is important, but I think the work that is done by Vincent Timmerman and Esther Wolves, where, as you call it, the fully human in vitro models are very important, because here we are taking a human protein, treating a disease that is relevant in a setting in a human context, but we have the problem of only having data in animals and in mice and potentially rats in the future. So as part of this work together, we want to look at common targets and mechanisms. And one of the things that we've been doing already is looking at macrophages and the involvement of macrophages. And we know that alpha-1 antitrypsin is able to switch macrophages from their M1 state to their M2 state. So this is known. If you analyze the gene patterns of macrophages treated with alpha-1 antitrypsin, we've shown this, that it's being nudged towards the M2 state. And just to really say thank you to my team currently in Geneva, and that our main objective is really to bring a therapy to patients as soon as possible. So I just want to say thank you to my team in Geneva, and really to leave you with really 3 key takeaways. If you remember nothing else from my talk today, I think what you should remember is that AAT has an excellent safety profile, that AAT inhibits ADAM17, which is a key regulator of myelination, and definitely is worth exploring further. And of course, AAT-FC is a better version of AAT. Okay, of course, it is an early development, and we are still a long way away from the clinic, but I think these 3 key takeaways I would like to leave with you today. Thank you very much for your attention, and I'm happy to take any questions.
Thank you very much for the very interesting idea and study. My question is, how good does it go to the peripheral nerves, this molecule, and do you know if it works also in the CNS? For example, for CNS myelination.
It's a very good
Do
question.
we know the biodistribution?
So we're working on CMT, but we've also looked at potentially deliveries into crossing the blood-brain barrier into the CNS. And there are now technologies of modifying the FC fragment or mutation in the FC fragment to bind either to the transferrin receptor or bind to the FC neonatal receptor. So we are working on a CNS version of delivering this into CNS by introducing certain mutations to cross the blood-brain barrier. And in terms of timelines, I can't really share with you exactly, but I think the mechanisms of getting it into kind of the stem cell niche and exhibiting its therapeutic effect is maybe similar.
Can you please explain, you did a treatment for over 2 weeks. Why did you stop? I mean, the therapy only 2 weeks. Was there a reason for the 2 weeks?
I get this question a lot, actually. In fact, we saw an effect after 2 weeks, but the reason why we had to stop, and this is another limitation of the animal models, is that you get anti-drug antibodies. It's a human protein in mice. After 14 weeks, it doesn't make sense to continue the therapy further, especially an anti-inflammatory, because you get antibodies to AAT. So we have to stop, and this is where I think the human models, the in vitro assembloids, are very important, is when we can study this for long-term. And I think this is the future in terms of long-term therapeutic assessment of human antibodies or human therapies such as ours.
Nick, great. How safe is it for kids?
Sorry?
For kids, for young patients, how safe is it? The drug or the protein?
There was a study, actually, in fact, that tried to repurpose alpha-1 antitrypsin for diabetes for children. There were some studies, and they went up to 120 and 180 milligrams per kilogram, which is triple the normal dose they use for replacement therapy. This was actually used these patients didn't have a deficiency. They already had the drug. They already have a normal level of AT. Absolutely no side effects except for the IV infusion, because these kids had to go in every week to get IV infusion, and they didn't like it, and they had some associated issues with that. But otherwise, no issues at all. So completely safe and completely, and as you would expect from a human protein, a naturally occurring protein.
Just one question. If it's safe and quite with a good efficacy right now, why are you developing a new molecule?
I'm the biggest advocate of taking CMT1A patients that are willing to participate and use currently available Alpha-1 antitrypsin for treatment. What we are developing is the next generation now. But it doesn't mean that Alpha-1 antitrypsin, if you see your doctor and say, I would like to try, maybe it will help, there's absolutely no reason why not. Where I'm just explaining to you what might be the next development after the plasma-derived one.
For the fusion protein, do you expect any autoimmune response? Far back. Because usually if you add mutations to it that are not present in the patient itself, there can be immune responses even to a single peptide.
So there is a whole science behind designing of FC fragments. It's not only our work, we're not the pioneers in it. Roche does most of the pioneering. What we did is we took what Roche has done up until now and looked at which patents expire, which one lowers the immunogenicity the most, and this is the ones that we introduced. So we are not big enough to do this kind of studies, but we took something that we can use and apply therapeutically straight away. So we are using something that is already in the clinic. So these mutations are already being used for antibodies for different diseases that are safe, effective, and we tested the immunogenicity in cellular-based assays to confirm that these do not lead to any upregulation of any inflammation in our
I think one more question by Mary, because then we don't have time for others.
Thank you for your presentation. It was very interesting. I just think, though, we cannot say to patients you should can go to your doctor and ask for it now, and a drug that's not been done a trial. I would just say we cannot say what you said, that you could go to your doctor and ask them to prescribe it now. As a clinician, I would not feel that's appropriate.
Sorry, I apologize. I will take it back. But I think the question is that there is no reason why necessarily this could not be used for CMT therapy, or initiated a clinical trial to test this therapy in patients. But sorry, I apologize. I didn't want to mislead in any way.